Hey class, this is lecture 15, gene expression, from DNA to protein. So we've had this conversation, right, about genetics and chromosomes and DNA, and what is the connection that takes DNA and makes a genotype into a phenotype? Like, how does that actually happen? So that's what we're going to talk about in this lecture. It's super interesting. So we talked a little bit about mutations, and here we're going to talk about, how changes in genetics can lead to changes in, phenotypes. So this like introductory example, how can one change in DNA result in such a dramatic change in appearance? So in the left-hand side, there is a donkey with a kind of normal brown color coat. And so the gene for pigmentation is represented here by the double-stranded helix, very simplified. And so we're going to talk more about the rest of this process, transcription and translation, and how mRNA makes a protein. But so this gene and this allele for the brown donkey color gets expressed. But one tiny change in DNA can make a whole change in these processes, and that's why there's a totally different outcome for this allele. This different version of this gene, this recipe. In this case, the phenotype is no pigment in the color, and so these donkeys are albino. There's no pigment deposited of the melanin, so that they don't have like a coloration. They're kind of just like white. And so this lecture will talk more about these processes, transcription and translation, and how that little change in DNA ends up with a totally different change. So we'll talk about how gene expression is the process by which DNA directs the synthesis or like making proteins. And it's proteins that are the link between genotype and phenotype. So the DNA inherited by an organism leads to specific traits, which is something that we've covered, right? And so this is like how. It leads to specific traits by dictating the synthesis of proteins. It's the recipe that gets passed on for how to make all these different proteins. So proteins are what are the links between genotype and phenotype. And so again, we're going to kind of like follow the story of discovering these things. So how was the relationship between proteins and DNA discovered? So we'll talk about that in a second, but yeah, in this whole lecture, we're talking about gene expression. And so gene expression is the process by which DNA directs protein synthesis. And there's two stages, transcription and translation. And so just as an overview, that's what we'll get into a little bit more later. But so how do they figure these things out? So in 1902, there was a British physician named Archibald Garrod who first suggested that genes dictate phenotypes through enzymes. He was like, enzymes do a bunch of stuff, so it must be enzymes that are what get made by genes. So he said that genes dictate phenotypes through enzymes that catalyze specific chemical reactions. So if you remember, back to like lecture 2 or 3, we talked about enzymes and how they're a protein that do catalyze specific chemical reactions, and you had that lab activity with the lactase enzyme. So yeah, each enzyme is in charge of catalyzing one specific chemical reaction. And so he thought symptoms of an inherited disease reflect an inability to make a certain enzyme. So, okay, this disease is because they can't make this one enzyme. Which, is a good hypothesis because cells make and break down molecules in a series of steps in a metabolic pathway. And so he thought, okay, if something gets messed up in that pathway, that must be the defect. So There was this group, George Beadle and Edward Tatum, who exposed bread mold to X-rays. And so I like this figure. So these bread molds were specifically of the genus Neurospora. So these are cells of the bread mold. And they are grown on what's called media. So when you grow bacteria or just small organisms, like molds, fungi, you grow them on like a specific kind of media. And so there's different kinds. And so when he placed normal cells on this medium and then subjected them to X-rays, these X-rays mutated the DNA, as we talked about last lecture, right? Different things can cause damage to DNA and cause mutations and maybe nucleotide changes. And so in doing so, he had some cells survive and then divide and form colonies. So these are the surviving cells after the X-rays. So they were able to survive, but then they were mutated a little bit. And this is totally random. So he just wanted to see, like, if you mutate them, what happens? Can we figure out what gene got mutated based on what ends up happening? So he took the surviving cells from these colonies and then put them on a different kind of medium. So complete medium has like a bunch of different nutrients. And so the cells are provided with everything that they need. But normal cells can also grow on what's called minimal medium, which means like there's just like the bare minimum there. But there's no amino acids provided or other stuff. It's kind of just like salts, glucose, and agar, and that's just like the minimal stuff. But because the cells normally can make their own amino acids, they're able to grow on this minimal medium. But when he took the surviving cells from these colonies and tried to grow them on minimal medium, suddenly they couldn't do that anymore. They couldn't make their own amino acids. And so he thought, oh, okay, that's interesting. Like, we did mutate something, and it was something in the metabolic pathways that made their own amino acids that we didn't provide to them in this case, and so they didn't grow. So then he took these mutants and then isolated them to different other kinds of media. He was like, okay, so if we know they can't grow because they can't make their own amino acids, there's a bunch of different amino acids. How do we narrow down which one it is? So they took tubes of media and had it minimal, but they just added one amino acid to each different tube. So this one was minimal medium plus valine, one of the amino acids, and that's it. Minimal medium in this one plus lysine, and that's it. This one only had minimal medium plus arginine, and so on for each amino acid. And so that when he did that and he grew or tried to grow them in each condition, if they did grow, then that means that that was that amino acid that it wasn't able to make on its own. So if it was able to be provided that amino acid, then it was able to grow on this minimal medium. And so just for reference, they had a control group. So if you remember back to lecture 1, it's really important in experiments to have a control group. Like, you know what this outcome is going to be, but it's not pointless. The point is, like, you know that this outcome is going to be like this so that you can compare it to other experimental groups. So for the control in this case, he had the wild type cells, meaning the normal cells, that were able to grow in this minimal medium, because these cells were not mutated. They were able to make their own amino acids, and so they could grow even on the minimal medium without any extra added help in added amino acids. So that would be a good comparison, because if the normal stuff grows and is expected to grow on the minimal medium, but if for some reason it didn't grow in this case, and that means maybe you made these all wrong. Maybe the minimal media like went bad that you're making everything from. Like, this is you're supposed to know what it's supposed to do. And so when you see the expected result, you're like, okay, I made everything right. And I could compare this to this. Okay, so let's go back to the slide. So that's what this group did. George Beadle and Edward Tatum, exposed the bread mold to X-rays, creating mutants that were unable to survive on the minimal media. So by doing this, they were able to see, okay, some of these colonies needed to be provided arginine, the amino acid arginine. So then their colleagues, Adrian Serb and Norman Horowitz, identified 3 classes of arginine deficient mutants. So there was 3 groups of mutants that were deficient in arginine, but there must have been some kind of difference. And so they wanted to see, well, why are there 3 different groups that can't make arginine? Are there different parts of this pathway to make arginine that aren't working anymore? And yeah, it turns out that each different class of mutant lacked a different enzyme necessary for making arginine. So, the results of this experiment, or these experiments, provided support for that first original hypothesis, the 1 gene, 1 enzyme hypothesis. Because, yeah, these enzymes were important in making either the final product of arginine or like the intermediates in this whole process to end up making arginine. So these experiments provided support for that, but then we learned more. So the hypothesis states that the function of 1 gene is to dictate production of 1 specific enzyme. And so this is kind of showing like the different enzymes I was talking about. So if this is the pathway to make arginine, there's actually 3 enzymes that need to be present to be able to make each intermediate. So if there's like a precursor molecule, enzyme A is in charge of modifying that into a different amino acid, ornithine. Enzyme B is in charge of the next reaction, which would be modifying ornithine to citrulline. And then lastly is a different enzyme that modifies citrulline to arginine. So remember, each enzyme is only in charge of 1 chemical reaction. So it's not just like 1 enzyme that does the whole thing. There's different intermediates. And so again, in the control, there should be wild type normal bread mold growing on this minimal medium. And so if you grew that and it did grow, okay, this minimal medium is made fine. I expected it to grow, boom. But if you grow the mutant cells on this same minimal medium and now they can't grow, then that's something that's wrong with the cells, not the medium. And so here are visuals of the different groups that they did to figure out, okay, which enzyme is missing in this pathway to end up making arginine. Because again, there were 3 different classes that couldn't make arginine on their own, but it might have been for different reasons. And, it turned out to be true. And so in all of these, this is the wild type control. And again, yeah, you really have to do it like every single time you do this. You can't just grow the control once and then say, all right, yeah, that's fine. Anytime you do an experiment, you have to run a control with the rest of the stuff in that experiment. When he grew stuff on the minimal medium with nothing else added, wild type grew, these other mutants did not grow. When he grew stuff on the minimal medium plus ornithine, a molecule, this grew, of course. It didn't really need extra ornithine, but it grew because it can make its own. In this case, only the class 1 mutants, like 1 group of the mutants, did end up having growth. So this was needed because the mutants couldn't make it on its own. And then these, didn't grow. So the same for each one. They were like, okay, these are different points in the pathway. After you provide this, they're able to make it. And so the enzymes must be needed later in the pathway. So then they identified like the different enzymes. So this class wasn't making enzyme A on its own. So if you provided the ornithine, then it was able to do the rest because the rest of the enzymes in that pathway were fine. And then same for this one. This one, it turned out it was enzyme B that wasn't working right. So if you provided this intermediate, skipping that step, then it could do the next step, and so on. So from studying this stuff, that was support for the proteins being enzymes. But later on, yeah, we learned more. Not all proteins are enzymes. So researchers later revised this hypothesis to be one gene, one protein. Maybe it's proteins that do other things, they don't all have to be enzymes. Not all proteins are enzymes. So researchers changed it. Okay, maybe it's just like the proteins. They don't need to be enzymes, but it's proteins that are necessary. So they came up with the one gene, one protein hypothesis. But again, if you revisit that lecture, proteins are actually huge molecules, and they're made of several smaller polypeptides. And so you might have several polypeptides working together to make one functional protein. We kind of refer to them like interchangeably, like a polypeptide is a protein, but actually it's more realistic to say, like, to specify, oh, this makes a polypeptide, and there's this many polypeptides making this functional protein. Yeah, so each polypeptide actually has its own gene. So if I could draw this, say there's like a bunch of, each of these makes a polypeptide. This is like a gene, and then this makes this polypeptide, this makes this one, this makes this one, this makes this one, and then they work together to be this whole protein. And again, remember, even though we show them as just blobs, it's actually more complex than that. It might be made of several polypeptides working together to make this one enzyme that we just represent as like a blob. So, therefore now, Beadle and Tatum's hypothesis is now restated as the one gene, one polypeptide hypothesis. So each gene makes one polypeptide, and then they can form together and create enzymes. Okay. So yeah, it's kind of more common to refer to gene products as proteins, but to be more specific, it's polypeptides. Okay, so we're going to talk as like an overview about transcription and translation. We'll go in more detail in part 2. But as an overview, so DNA and RNA are nucleic acids, and we talked a little bit more about DNA in the last lecture, but RNA is very similar. So again, it might help to go back to the macromolecules lecture. But so RNA is like the bridge between the genes of DNA and building a protein. So transcription is making RNA using information in DNA. And what transcription ends up making is an RNA known as messenger RNA. And so that messenger RNA can then be used in translation. So there are many different forms of RNA that we'll talk about, and one of them is messenger RNA. So translation is the synthesis of a polypeptide. So you're building a polypeptide using information in the mRNA. And this happens at ribosomes. Those are the sites of translation. So in prokaryotes, translation of mRNA can begin before transcription has even finished, because remember, prokaryotes don't have membrane-bound organelles like a nucleus. And so the DNA is just like in a region of the cell. And so ribosomes can be pretty nearby. So you can like translate and transcribe like at the same time. And I think that'll make more sense as we go through the lecture. But yeah, that's an interesting comparison. Versus in a eukaryotic cell, because there is a nucleus, that nuclear envelope separates transcription from translation. So this process has to happen here in the nucleus, and then this process has to happen out here. And so after transcription, eukaryotic RNA transcripts are modified through RNA processing to yield the finished mRNA before translation happens. So this is just a lot of words. Maybe it's not helping yet. But this is kind of like the big picture. So this is gene expression, basically. There's DNA, and that is what dictates the RNA that's going to be made. And then that's what dictates protein. So DNA is read and gets transcribed into RNA. And so that is what, our textbook calls a primary transcript. It's like the first draft. And so that's the initial RNA transcript from any gene prior to processing. Then you kind of have to fine tune it. So this process is overall called the central dogma of biology or of molecular biology. So a dogma is like a belief. So this is like a core belief. This is a fact. So this is the concept that cells are governed by a cellular chain of command. DNA to RNA to protein. Transcription is that synthesis of RNA using information in DNA. And yeah, the first draft is the primary transcript, or also known as the pre-mRNA. It's the initial RNA transcript from any gene prior to processing, just basically what the previous slide just said. And then after that is translation. So it's the synthesis of a polypeptide using information in the mRNA. And so this is an analogy I came up with that I think really helps. So if you think of DNA as this enormous cookbook, so my sister got me this cookbook called The Joy of Cooking. It's enormous. This picture doesn't do it justice. It's this thick. And it doesn't just tell you recipes, it's also how to fillet a fish, how to debone a chicken, how to whatever. It just is everything you could ever need, basically. But there's a lot of information in there. And so because I don't feel like buying a new one, I'm going to take care of this, right? So that's kind of like DNA. Your DNA is all of the recipes, this huge recipe book, and you can basically make anything you want from it. But you also want to take care of it because you don't want to buy a new one, or you can't buy a new one. And so the recipe book is DNA. But when you want to make something, you only need one recipe at a time, right? You don't necessarily need the whole cookbook. You just need one recipe. And so if you were to copy down a recipe onto an index card or a notepad or a Post-it note or whatever, you have one little temporary version of that recipe that you can take into the kitchen. And it doesn't really matter if it gets spilled on or torn or burned or whatever, because it's just a copy. You can go back to the book and make another copy, right? So RNA is kind of like that. It's a small temporary version of a recipe for something that you want to make, a polypeptide, that can leave the nucleus. It can maybe go get damaged or whatever. And then once you're done using it, you could just, crumple it up and throw it away. So RNA is a temporary recipe on an index card or a Post-it. And then the thing that you're making, the end product of what you're cooking is a protein, or again, a polypeptide. And so if you are thinking of Thanksgiving dinner, for example, how to cook the turkey or whatever, Yeah, you don't need this whole book for everything else yet. You just need the turkey part. So you write down steps, and then you cook up your turkey, and boom. So that is like a good, I think, analogy for gene expression. A newer one I thought of recently was 3D printing. So 3D printing, you have a final product, your printed whatever. You also have a code. So you put in like a specific code, you look it up or whatever, you put in a specific code and that 3D prints that one specific thing, like a Batman mask for your cat or whatever. A bottle opener, whatever it is, like you have to put in a code for that specific product. And so I guess in this analogy, the rest of the DNA would be like the internet where you could find all the code, or maybe your brain, you're thinking of like how to make this and you happen to know how to code certain things. I don't know, whatever. I don't know about 3D printing that much, but so that's another analogy. And again, based on what kind of cell, it's going to determine where this happens. So in a prokaryotic cell, like a bacterial cell, DNA is not hidden behind something in a nucleus. It's kind of just free floating, right? And so all these things can happen at once, basically. But in a eukaryotic cell, there is a nucleus dividing some of these steps from each other. So DNA is always going to be in the nucleus, and so transcription has to happen in there. RNA processing, where you kind of fine tune that recipe a little bit, happens inside the nucleus too. And then RNA is kind of safe to leave and go be translated in the cytoplasm. Okay, so I think I'm going to pause it there, and that'll be part 1.